My name is Lauren Pang. I'm the health officer for Maui County. I've been the health officer here for the last 22 years. Today is April 29th, 2022. We're at a tail end, or hopefully tail end, but we're still having COVID. Now, I am going to give a presentation which I've been dying to give, but I was never quite allowed to give because I was challenged on this issue that I was spreading disinformation. And that had to be appealed. And during the appeal process, the last 7 or 8 months, my lawyers and people told me, don't say anything. I really wanted to explain this on my side of the truth. It involves off-label treatment, or also called repurposed, the slides also recalled repurposed approach to product development. So the title of this talk is more than just COVID. It's more than just one product. It is the regulatory product development process in medicine. Now, most things, products, have a regulatory process, like tires for a car. You want to sell tires, you got to prove it works, you got to prove it's safe, you got to prove its consistency, and it's not dangerous. That's product development. But in medicine, we develop products too. And there is a regulatory process. So here's the title, yep, in medicine, yeah. And I'm going to focus a little bit to tell you where off-label treatment or approach strategy fits in. And a lot of people in medicine call it off-label, but the public, we like to call it repurposed. So you made the product against one disease, and you find out that the same product, same dose, same formulation, might be useful against a different disease. So it's repurposed for a new disease. That's an interesting strategy in our overall product development. Here's me, Lauren Pang, MD. I have a medical degree from Tulane University, and at the same time, I took my electives in public health. So I have an MD from Tulane University. I will speak as a private citizen, but in this topic, I can speak as the Department of Health person. But before I took this job in the year 2000, for this period and overlapping for 5 years, I was the World Health Organization United Nations consultant, mostly in malaria, but in many other infectious diseases. And for that period, I was also with the Walter Reed Institute of Research. Okay, let me advance the slide. So here I was, working for a long time in product development with the World Health Organization. I'll paraphrase what I have written on the slide. So we're not just going to read the slide word for word, okay? In the area of repurposed, here it is, repurposed products, all right? I've worked in this area for decades. I've taught, I've published, I've worked with the other developed countries trying to harmonize the strategy. So we all have the same criteria. I have learned how to analyze data on all kinds of different studies, and I even took it, once I came here to Hawaii, to apply the same analysis and principles to non-drugs, to environmental issues. For example, smoke, cane smoke. Well, if cane smoke really affects our health, you would expect the higher amounts to affect our health more. That is called a dose ranging study. We learned that from drugs and vaccines and diseases, but I can apply it to environmental issues, higher dose, okay? Some people said that I have been doing this a long time, and maybe I'm doing it poorly now that I've lost my marbles. I agree. I don't think somebody who has done something great in the past, won a Nobel Prize, gets an automatic pass to say what he wants now. We should judge them for what they are saying now. It is true. What he has done in the past might apply to things now, so he might call up historical evidence of what he's done, but only as it applies now. So if you're going to say I'm giving disinformation now, let's hear what I have to say now. Conversely, let's not say that he did these great things in the past, so he gets to speak now. No, maybe I'm losing my marbles, maybe not. So let's look at this, okay? So I've spoken as a private citizen on many issues in the last 20 years when I came to Hawaii. Cane smoke, pesticide drift, flesh-eating bacteria, dengue eradication, depleted uranium, 5G, the communication network. I was always allowed to speak as a private citizen and asked to be judged on what I said, whether or not it aligned with the different agencies. Sometimes it did, sometimes it didn't. But on COVID, I was recently, several months ago, said I was giving misinformation and I was not allowed to speak as a private citizen. Those allegations have been cleared. So now I present my scientific argument. Again, I think it's truthful. I still don't know, do I speak as a private citizen or from my department? I will just ask the public to please listen to what I have to say before deciding who I should or shouldn't speak for. All right, the next slide, please. Product development. In the medical field, the products are drugs, vaccines, and diagnostics. Already some, obsessive people might say, well, no, that's not the only product. There's procedures, different ways to do the appendectomy. Okay, so yes, I did leave out procedures, but in general, drugs, vaccines, and diagnostics. These are the areas we worked in medically for developing a product. Pick a product, say a drug. Okay, just for example, say a drug. I'm going to refer to drug from now on. I want to know its safety. I want to know its efficacy or effectiveness. Does it work? And I want to know in general, whatever you did to show me safety and efficacy, can you make sure I can repeat it? That your observations are replicable, repeatable. It's kind of like baking a cake. Yeah. I want to know if your ingredients are safe. I want to know if it's effective. Do people like it? Is it delicious? And can you replicate it? So can you give me the same recipe? All right, next slide. When we go about developing a product, safety, efficacy, replicability, we were taught repeatedly the mnemonic, GMP, GCP, and GLP. Good manufacturing practice, good clinical practice, good laboratory practice. So whenever we mess with these products, make sure you have GMP, GCP, GLP. GMP, good manufacturing practice. When you make the drug, okay, it could be a vaccine, it could be a diagnostic test, but I'm just going to use drug as a substitute for the products. When you make a drug, can you do it consistently? That is the potency, the dose variation, contaminants, the purity. So the two contaminants, general types are chemical contaminants or biologic contaminants, viruses, bacteria. Are you making it for delivery orally, injected, suppository, inhaled? Can you do the delivery system consistent and stability when you keep it on the shelf? All right. Now, this is true for animal products or human products. Make the product consistently so when we study it, recommend it, if we recommend it, and approve it, you know what you're dealing with. Next, GCP. When you test these products for safety and efficacy and replicability, and it involves patients. C, it's clinical. I didn't actually know this in medical school. As soon as you say clinical, it means humans. Okay? So in your patients, when you study the patients, did you have a good idea of who you included, who you excluded, how you monitored them, what the outcome or outcomes was, and was it maybe a randomized trial or not a randomized trial? Who were your controls? You're comparing it to what? Not taking the drug or standard therapy? And were there biases? In your study, did people know who got which drug so you could kind of fudge the results subconsciously or consciously? That's GCP. Then there's GLP, good laboratory practice. What lab are you using? Lab for what? Well, you are going to test your formulation. Whoever made the product, I would send it to the lab to check. Once again, dose variation, the actual potency. I would check it for purity, chemical contaminants, biologic contaminants, et cetera, et cetera. And then Oh, sorry, the picture cut it off a little bit. But you will run the lab on your patients. Really? Yep. When I do patients, I need my lab. Why? Don't you just give him the drug and see if he cures? Well, there's a couple of things you do the lab on. Sometimes the marker on the patient, his blood levels, his liver levels, that requires a lab. Was it a good lab? Was it a certified lab? But more importantly, when the drug works or it doesn't work, and you did all your liver function lab tests, I kind of want to know why it worked or didn't work in him, why it worked or didn't work in Brazil versus Bangladesh. And so you would draw blood samples to check your blood levels that the patient got. I know he took a good formulation, but what was his drug levels? And then we also draw a sample of the germ, if he didn't cure, or the germ just before he cured, to see what strains of germ we are dealing with. And here it is in the slide again. This is very important. If I can draw the germ isolate on a cure or non-cured patient, I could grow it up in the lab and test it in the lab against drugs to say that it's a confirmation that not only did he fail clinically, but the germ that he broke through with, it failed in the lab. You have a resistant strain. Okay? So GLP, GMP, GCP. We use it through the whole process. Oh, by the way, look at this picture here on the slide. You see some formulations, GMP, are for horses and some are for humans. That's automatically a problem because if the mean dose is the same for horse and humans, the dose variation might be very different. So if you go dose 100 people with the horse medicine, it's all over the board, and so a lot of them are going to get toxic from too high a dose or not work at all from too low a dose. So the dose variation is just as important as the mean potency. Okay, next slide. Now this slide is pretty busy, but I practiced giving this to medical people and they said, that's your best slide of all. Well, for the general public, I'll try to keep it a little bit entertaining at least. So here we go. You have GMP, GCP, and GLP. It applies to all aspects, all phases of your development process. Okay, so we call the development process phase 1, phase 2, phase 3, phase 4. And you are to do the phases stepwise. You're only going to move to phase 2 if phase 1 was successful. And you're only going to move to phase 3 and 4 if phase 1 and 2 was successful. So you do it stepwise, okay? But before you even approach the clinical phases, clinical is human, don't feel bad if you didn't know that. I didn't know that. I think it was until the second year of medical school. You are going to do the preclinical stuff, non-human. What are you going to do? I'm going to the laboratory, I have cell cultures. I can go to the laboratory, I have mice. I have any animal I want in the laboratory. Now, recently, we kind of have a moratorium on primates because they're endangered. Okay, fine. But in the laboratory, I am going to test my products for safety, efficacy, or effectiveness, and replicability in case somebody wants to repeat it. So here's a cell culture. You grow the germ, you dose it with the drug. Well, it works or it doesn't work. That's how we pre-screen some COVID for early treatment. We just went and tested some drugs off the shelf for other purposes. We can do it in culture. If you can't culture it so well, take it to the animal model. Do you have an animal model for the germ? Yeah. So you get 100 mice, you dose them with different drugs to find efficacy. How are you going to do toxicity? Screen the drugs for toxicity. Well, I don't think the cell culture is going to tell you this is going to be toxic in humans. The mice might. They might get lethargic and weak and go into coma. I think it's toxicity. Are you sure? It wasn't the disease. It was the drug itself. Well, here's a bunch of mice. I didn't even give the disease. You dosed them and they got toxic. Or here is an animal model, the sheep. This is what my wife and I used because the issue was in antimalarial drugs, arrhythmia. The sheep is a very good model for cardiac arrhythmia. So here you are in the lab doing all the stuff. And when that finishes and you kind of know what you're going to do, efficacy, and safety. Then you move on to the humans, phase 1. Phase 1, small groups of humans, maybe 5 to 7 per group. They are uninfected. They're healthy subjects. They don't even have the disease. You're going to stepwise give rising doses of the drug, rising doses, lower, middle, little higher. And you are looking for toxicity. Your job is to find the highest safe dose. You mean you keep dosing till you get some toxicity? Yes. And you are going to, on your way to find dose X, the highest dose, or when you start to get toxicity, the highest safe dose, you're going to find for me the target organ. I don't know why they call it organ, but that's the marker for the first sign of toxicity. It could be a sign, a laboratory test on urine or liver or blood, or it could be a symptom, like I think I'm having a headache. Okay, so that's your first sign or symptom. For my purpose, I call this TSS, toxicity, sign, and symptom. Okay, that's all you want. From rising doses, small groups, looking for the highest safe dose, and on the way, your first indicator of toxicity. If that works and you know what you're doing, you got those two things, you get to move to phase 2. Phase 2, small groups again, 5 to 7 people, sometimes 10 or 12. These people are infected. In general, they're healthy volunteers. Okay, so now your job is to give rising doses to cure the disease, whatever your outcome is. I need to know the dose that'll cure people, and you will stay below dose X. You're not going to give them the toxic dose. So hopefully you can start a little low, and you're going to give rising doses till you cure. And you're going to find the lowest curative dose, the lowest dose that can cure. Call that Y, dose Y. On the way up to dose Y, the lowest dose that will cure, you're going to watch for TSS, toxic signs and symptoms, and I call that TSS prime. Because you might see toxicity, the target organ, at a lower dose than X, because you have the drug and the disease. So when the two overlap, oh my goodness, you're going to see toxicity early. This is very tricky. That's why we often have doctors, there's nothing magic about a doctor. Sometimes the nurses pick it off first. The guy is getting sicker earlier, and we haven't even reached dose X, Dr. Pang. Oh, is that from the disease? Or is it from the drug? Or is it from both overlapping? I don't know, but we record that. When you get phase 1 and 2, now you know dose X, and now you know dose Y. Hopefully, dose Y is below the safe level or the toxic level. For the medical people, we call this the therapeutic index, X over Y. Rather than belabor it, let me give you an example. The curative dose Y starts at 5 units. The toxic dose starts at 9. So we take 9 over 5. Or you can take 9 minus 5 if you want. You see, the toxic dose is higher, 9, than the curative dose. That's good. That's a high therapeutic index, 9 over 5. Anything better than 1 is pretty good. So you got a lot of leeway. You know that dose 5 that cures? Well, you could have given him 6, 7, or 8 before he got toxic. So that's good. The toxic dose exceeds the curative dose. Well, why don't we just give him 8? That'll really cure him without getting toxic. Because people differ. Some guys get a little toxic at lower doses, and some guys need a little higher dose to cure. So I like to have a little margin of error. Okay? And once you get phase 1 and 2 figured out, oh, by the way, what if the therapeutics index is greater than 1? He reaches toxicity before he cures. Oh my, should we never do this? Depends what disease. If it's cancer, I think the guy can have a little side effect, headache or nausea, because you're trying to cure cancer. So we're willing to deal with a little lower therapeutic index when the disease is pretty severe or you don't have alternative treatments, okay? But in general, let's say you got a decent therapeutic index. You got room to increase the dose. And you do the phase 3. What's phase 3? Large numbers, infected people, well, I put infected people, randomized and controlled. Wow, large numbers. You do that for statistical certainty of your toxicity, TSS prime, and your curative effect, large numbers. It's kind of like somebody says, this drug works. How many did you try it in? 3. Don't you think you should try it in 30 or 300 for a little more statistical certainty? Yeah, I think so. Okay, fine. Statistical certainty, and it's randomized, so there's no fudging. Often I've seen it. People would give the new drug to the sicker people because, well, that's what we're trying to do, right? Get a new drug. You shot yourself in the foot. You gave it to the sicker people, and the controls weren't that sick. So, of course, the two things look the same, the controls and the drug, because you gave it to sicker people. Next time, randomize it so they both have the same level of severity, et cetera, et cetera. Trust me, we've aborted 2 years' work, $200,000 in developed countries because people did not randomize. They did not understand why they should randomize, but they did not, whatever. They said they would, but they didn't, okay? If you pass phase 3, you still have a good therapeutic index. You have statistical certainty with big numbers of the TSS prime, the toxicity in diseased people, and the curative effect, then it's approved. Then it is approved. Got it? When it's approved on label or for a label. You get the approved drug, you take out the label, and it says you are to give this dose, specific dose, against this disease. So on label means this dose, this disease. Off label means, to me, this dose against other diseases that we don't know if it works. Now, the medical guys know this because on-label means, what did the package insert say? Off-label means stay below the safe dose. Go ahead, try it on another disease. But make sure you know what you're doing because you might see TSS prime. And what was the alternative treatment anyway? Did that work? Then why'd you guys give this thing off-label that it might not work, okay? But for the general public, they said, we like to say repurpose, purpose for another disease. And this is kind of important to me. After phase 3, and it's approved by the FDA, then we take it to phase 4. What is that, phase 4? It's approved for the general public, so we release it to the general public. And in the general public, you will see all kinds of variations of people. They have comorbidities, they're taking other drugs, for their high blood pressure, taking insulin for diabetes, they're taking asthma medication. You're looking at drug-drug interactions. They are taking it for all severities of the disease, not just early, they're just kind of late. You told them to take it in the first 5 days of illness, and whatever. They took it in the last, after the first 9 days. They didn't get to the drug. Sorry, but that's how they took it. They know how they're supposed to take it, but they didn't, they just couldn't. There's a subtle point. Efficacy is the curative effect when taken as prescribed. Effectiveness is curative effect when taken as they kind of had to do. They tried to do efficacy, but they didn't get the drug. It was a long trip to the pharmacy. The pharmacy was closed until later. In the phase 4, you will also see strange things happen. Missed dose, supposed to be 3 times a day for 7 days. You only took 2 days? Why? I was feeling better. Then there was something worse than missed dose, overdosing. Why would the guy overdose? Well, simply this. It's a 7-day course of therapy, 3 times a day. After 2 or 3 days, he felt better, so he stopped taking it. Why did you stop taking it? Well, it was expensive, and I had to go 5 days to get the medication, and I saved it for my next bout of malaria, and then I gave some to my brother who has the same symptoms. Okay, that's understandable, okay? But on your 5th day, you began to get sick again. So what did you do? Well, you know those days I missed? I doubled up. I took the day 6 and day 7 dose, but I took the 3 and 6, and I doubled it up to catch up for the missed doses I took. Don't lie to me, just tell me what you did. Oh, no wonder you're toxic. You took too high a dose. Oh, you mean just doubling up the dose is going to make me toxic? Well, it depends on what the therapeutic index is, my friend. We are running these drugs so they cure without being too toxic, and you doubled up a dose. Okay, thank you, doctor. I understand now. All right. So when the phase 4, when people generally use it as they use it or abuse it as they abuse it, we are out there hunting out why it failed, why it was toxic, and we have this huge, vast knowledge of this drug for that disease under all these strange conditions. Okay, phase 4. Not mandatory. It's approved after phase 3. Okay, so that's the most painful slide of all. You remember the medical guys liked it. They said that's the most informative. So when drugs are not available, they're just too early. We have a new pandemic. I have no idea how to treat this. Or the existing drugs fail, or they're too toxic. Then we look at repurpose or off-label strategy. Repurpose. Repurpose. Let's call it repurpose. You start at phase 2. You know the toxicity. You know the highest dose X because it was used for another disease. So you know that safe dose X from the approved use of another disease. You know the target organ to follow for toxicity. So we can just start with phase 2, efficacy against the new disease. Phase 2, I won't flip back the slide, but you remember it is a rising dose looking for the curative dose, the dose Y. That's how we do it, dose ranging. Changing the dose, looking for curative effect. Okay. When we enter phase 2, it's under informed consent. It's still experimental, not for safety. Well, kind of, if it's TSS prime, but we know TSS under the other disease, and we kind of boldly extrapolate the safety from one disease to another. In my original presentation, I gave a warning about TSS prime, the drug we felt comfortable with chloroquine for malaria. We know the target organ from too much chloroquine, and it is arrhythmia. We were treating COVID, and severe COVID, it turns out, after all the autopsies, they were having arrhythmias because they're throwing embolic clots to their heart centers. So I gave a warning, I did, that when you use something off-label, chloroquine, which is safe for malaria, when you combine it with severe cases of COVID, you might overlap the arrhythmic potential. But anyway, that whole thing was interrupted because I was considered giving misinformation. Okay. Now, here we are giving phase 2. You're looking for efficacy by changing the dose ranging. You notice you're not doing a placebo yet. You're going whatever standard therapy is for COVID or whatever your new disease is. You will operate by GMP, GLP, good medical practice, good clinical practice, good laboratory practice, okay? And that's why we expect doctors to do it. A doctor doing a phase off-label or repurposed trial, a doctor would automatically say, what's the formulation? Because how in the world would a doctor use a horse medicine and extract the highest safe dose? Because I know it's dose X, but the variation on horse medicine is so large, you might just by picking a dose of horse medicine, give them a much higher dose. How in the world could a doctor replicate a study if the dosage of a horse medicine is all over the board? How could you extrapolate from a successful phase 2 study of a horse medicine to a phase 3 study? So obviously, to me, the formulation is so important in efficacy, safety, and replicability, because you're going to move phase 2 to phase 3 from an off-label drug, which we often do. How in the world would you do it on a horse formulation? So I'm a little bit insulted when they said that, he's doing the horse formulation. I am not. I've never done this. And I've explained many times since the year 1985 why we don't do this, based on the principles of safety, efficacy, and replicability. Okay. So that's phase 2 repurpose. And I told you this is all in product development. Product development has a phase 3, the big randomized controlled study. That's kind of like the gold standard to many physicians. Yeah, okay. That's a gold standard, or the best we can do. That's the end stage. But itself, a phase 3 study, is it possible to give discrepant results? Not necessarily wrong results, but discrepant results. For example, we do something off-label. It seems to work. We move to the phase 3. We give it big studies. One in Brazil, one in Bangladesh, maybe one in Pakistan. Big studies, treating people. Is it possible that 3 studies give 3 different results? Well, if you read the literature now, we seem to be at each other's throats. It doesn't work. It does work. It doesn't work. It doesn't work. You're giving misinformation. You're giving misinformation. I will solve this and show you what is going on here, and that practicing GMP, GCP, and GLP blindly, if you really didn't know what it was for, it will solve this issue of the phase 3 discrepant results. Okay, next slide. In summary, summary. The above things I talked to is an approved US, FDA, and World Health Organization process. Okay? A product is given approval after phase 3, and it's for a specific dose for a specific disease. Phase 4 post-marketing adds to the knowledge base of safety and effectiveness in all kinds of patients, in all kinds of situations. The strategy of product development emphasizes safety first above effectiveness or efficacy. It allows that safe doses in a repurposed drug can be extrapolated from an approved indication from one disease to another. It skips the need, repurpose, skips the need for phase 1 toxicity, which is a huge hurdle, and all that preclinical animal toxicity. It jumps right to say phase 2. It saves a lot of work. It has proven to be a valuable strategy, repurpose in many, many, many repurposed products. And then we take it to phase 3 and phase 4 after that. Okay? When you have phase 4 on an approved product it's widely used for decades after approval, like ivermectin or chloroquine, you have added safety data because it was looked at in a variety of settings. So never underestimate the value of phase 4 studies. Okay? And here's an example, ivermectin. It was approved for parasitic infections, not COVID, not yet, maybe not ever, but it was approved for parasitic infections, river blindness. Okay? And now people are trying to take it repurposed to look at COVID, or at least we did 7 months ago. The physicians carefully watch for new safety issues because COVID plus, remember TSS prime, there it is, might have interactions, okay? But don't forget, this ivermectin or chloroquine, whichever you want to use, is phase 4. We have decades of experience used in strange settings with strange self-administrations and strange underlying comorbidities, strange other things, okay? And we simply use a dose-ranging study to look for efficacy, phase 2. I will give you examples, no more theory, well, not too much, of phase 2 repurposed drugs and phase 3, why big randomized trials give different results, that we shouldn't claim the other guy's wrong or the other guy's right or whatever. Phase 2, dose ranging. Dose ranging was also phase 1. You're changing the dose looking for toxicity. Phase 2, you stay below that safe dose. You're changing the dose looking for cures. Okay. Phase 2 studies. You give me a case, a bunch of cases, large numbers or small numbers. They're not randomized. They're not controlled. There's no placebo. So it's not a randomized control trial. That's phase 3. You just give me a lot of patients, okay? I know the safe dose, either from phase 1 in a new drug, or from the approved use in another disease. Okay. The goal is to determine efficacy using dose ranging analysis. Okay. And remember, repurposed drugs start with phase 2. Here's a good example. I won't read it for you, but there's 7 doctors. They're treating COVID with ivermectin. They follow the FDA guidelines, stay below the safe dose, approved from the parasite study, the river blindness study, yeah? And they say, oh, by the way, Dr. Pang, there were decades of phase 4 study for ivermectin. Yes, thank you, okay? The medical doctors conduct it. They're looking for TSS prime. We are not using a veterinary formulation, and it's informed consent, telling the patient, we don't know if this is going to cure you, but it should not cause toxicity because we know the safe dose. Careful. When you give informed consent, a lot of patients will say, so you don't know if it's going to cure, it just won't hurt me. And what's the standard treatment? Well, there is none, or we don't have any yet, or it's too toxic. Okay, so I'm willing to try it. But doctor, after you get my data, you're going to figure out for the rest of the world, and for maybe my family, and for maybe me when I get COVID again, you're going to figure out when it works and how it works and when it's too late to give and what dose to give. You're going to figure out why and where it's efficacious, right? I'm contributing to medical science, maybe to myself if I get another COVID infection with another variant. Yes, I agree to that. Okay, here's the 7 doctors. They each have their little case series of off-label treatment. You know, these are different numbers of patients they have. It adds up to 80. This is an example. I needed 80 because it's round numbers. These doctors bring them in. There's no conflict of interest. There's no coercion. They come in. They give some case definition, who we're going to bring in, who's not. They give some outcome, don't die, don't go to hospital, don't go to ventilator, cure better, whatever it is. It could be a slew of outcomes. And then they're going to look at risk factors. So why did they cure? Is it age? Does age affect the cure rate? Is it the level of illness? Is it too late to give the guy treatment? Is it the promptness of therapy? 5 days? After onset, you treated them within 2 days, 3 days, 4 days, 7 days? Is it the promptness? Is it their weight? What do you mean the weight? Well, I gave them the pill, but each guy has a different weight. So the dose per weight changes. You just weigh the guy, right? Et cetera, et cetera. Did they get prior vaccination? I don't know. Were they having comorbidities? But all these risk factors might affect your cure. So you take all these 80 patients, and I do what the statistics people call trend analysis. I'm going to look at the outcome ranking by these different variables. I rank them, say in quartiles. You know, the upper quarter, the mid, the next quarter, the lowest quarter. The highest, whatever, 25%, then the 50, you know, it's quartiles. Let me choose the quartile, the ranking by dose. Wait. By dose, didn't you give them the same pill, the same number of pills? Yeah, but I divided it by weight. They have different weights. So I can rank it. Here's the quartile. Very low dose, number 1. The low dose is number 2. Medium dose is 3. The highest dose is 4. Okay. I just simply rank them. Here it is. Here's the outcome. Here's the 80 patients, yeah? 20 in each group because I quartiled it. Yeah? Here's failures. Here's successes. Here's my ordering. 1, 2, 3, 4. Let's say that this rank number 4 was the best thing, the highest dose, the earliest treatment, the youngest age. And number 1 is like the lowest dose, the latest treatment, the old guys, you know, like me, who are not going to do so well. So I just rank them, and then I put in your data. Each group, because it's quartiles, is 20, yeah? Here's a cure rate, 2 out of 20, 4 out of 20, 5 out of 20, 9 out of 20. These are just hypothetical numbers, okay? You see that the better the ranking, the higher the cure rate. This is called trend. there's a strong trend. And this p-value to the medical people means, whoa, you showed that with the better, that this treatment works either by a higher dose, or if it's not the higher dose, maybe it's the younger age, or maybe it's the earlier treatment. Oh, see, I do one of these tables for each ranking. One for age, one for promptness of treatment, one for dose. Although you gave them the same number of pills, you divide it by the weight, right? Or maybe your results are like this, 20 in each group, and there's a cure rate here. I simply put this one here. And so the cure rate was okay, but then it goes down with whatever dose or promptness of treatment. And then it goes up, and then it goes down. So there's no trend here. Usually when there's a trend, that means you're onto something. The drug's kind of working. You notice there's no placebo. I don't compare this to a placebo. I compare it amongst the doses, like a phase 1 and phase 2. That's a dose ranging. So this is how we do dose ranging studies, jumping into phase 2 for a repurposed drug. Okay, that's all I wanted to talk about phase 2 and repurposing drugs. It can be done. It's been done. I've done and published many times on this in the past. Okay, phase 3. The day the medical board cleared me, they found no purpose to look further. I did not give this information on phase 2. The media said, oh, and by the way, there was a phase 3 study on his ivermectin in Brazil, and it failed. Whoa, was this supposed to be a counterpoint? That when it failed in a phase 3, too bad, in hindsight, everything he said about phase 2 repurposed drugs was wrong? Absolutely not. I did not get to speak until now. In a phase 3 trial, remember, large numbers for statistical certainty on toxicity and efficacy. I get it. It's randomized, so we don't have bias. Yeah. It's blinded, so people don't say, well, give the new drug to the sick guys, because that's why we're doing new drugs, right, Dr. Pang? No. In the end, you hurt yourself, okay? Still, what could go wrong in the gold standard phase 3? Why might there be differences in the phase 3 trials? Okay, let's get something real clear first. Here's a publication, and I take it that they are honest. In 2015, the Nobel Prize was given in medicine. It was for the discovery of ivermectin against a disease, the actual effect against a disease, not the basic science, not the animal cultures, not the in vitro cultures. They hadn't done something like this in 60 years. And it was given against a human disease, okay? There, given for the human disease, not given for the horse treatment, not given for the horse disease. Yes, it does treat horses for their certain parasites or their certain problems. But it was given for the treatment of human disease. It was developed, actually, start development for the human disease, but it reached the horse approval first because the humans were being very safe and we did not have a good animal model to try it in. You mean preclinical animal model? Okay. So why do they keep showing horses? I have no idea. Physicians supposed to be running phase 2 pre-purposed drugs. You could not approach it because it's not replicable because of the dose-dose variation in the horse, because you don't know the target organ in the horse. How do you know when the horse says, I have a bad headache? I know when a human does. Okay, so to me it's a little bit insulting when they keep running this thing as if it was only a horse medication. And then in the same article right here, 6 out of 7 meta-analysis showed ivermectin in the randomized controlled trial, phase 3, found notable reduction in COVID fatalities. Wait. What's a meta-analysis? Don't worry about it. When I joined WHO in the year, when did I go there? 1992, we were stumbling over that term itself. Meta-analysis simply means you get all the big phase 3 trials you can, and you analyze them all together. It's like a big group study, and you look at them carefully and you say, all together, these phase 3 trials, either 14 or 15 or 25, whichever phase 3 trials you wanted to choose, it shows benefit or harm or whatever. 6 out of 7 showed benefit, reduction in COVID fatalities. Oh, and 1 didn't. Oh, so what? 6 says yes, 1 says no. Are we going to fight? The people who did the 6, are they going to say the 7th that showed nothing is disinformation? Are we going to say vice versa? The 1 that showed nothing, are they going to say the other 6 is disinformation? I don't think so. I think you're both right, but we have blurred the issue because of a failure of GMP, GCP, and GLP. Here is the study on the day I was cleared, put in the newspaper, well, Brazil showed it didn't work. So there, Dr. Pang. The Brazil study in New England Journal of Medicine. New England Journal of Medicine. Hey, that's a good journal, right? Read the article, find out that the Brazil study, who did they treat? They treated these patients. This is time. You can't see it, but that's April 2021. And this goes to October 2021. And on this axis is the percent of the variant circulating at that time. So in this period, whatever it was, the period of the study, these were their patients. Almost all of them were the pink variant. What is the pink variant? The pink variant is called P1, alpha, beta, gamma. It's the gamma variant. It arose in Brazil. It's peculiar to Brazil and the neighboring countries. So in your study where it failed, in the New England Journal of Medicine, you are testing it against the pink variant. In a study, another study where it's worked, Bangladesh, you did your study, I don't know, it was before the year 2020. That's off the chart in Brazil. What was the variant? I don't know, but it was not the pink variant. I don't think Bangladesh ever had the pink variant, even after it emerged in Brazil. So you are looking at variations in the germ. Could that be the reason why it worked in Brazil and failed in Bangladesh? Yes, it could be. You could be. Well, gee, Dr. Pang, you don't really know. You're grasping at straws. That's true. It could be, though. And I don't think we should challenge each other too fiercely. But there's one problem here. Part of GCP, GLP, and GMP, you are supposed to go and get the drug levels in these 2 areas. And you are supposed to get the isolates in the people who have failed, like 90% of the Brazilians. You're supposed to draw the germ and take it back to your lab to culture it, to challenge it with your drug, I guess ivermectin, or if you were testing any other drug, or your monoclonal antibody, if you want to do it, to say that this confirms, kind of, that what failed clinically, I drew the isolate and I cultured it, and it confirms it did or did not work in culture. You draw the drug level because maybe in Brazil, it was horse medicine, I don't know. Maybe it was good medicine, but sat on the shelf too long. I don't know. But you did not drew the confirmatory stuff, and you should have done it in Bangladesh. So they didn't do this. At least it wasn't published that they did this. Maybe they did it and are going to tell us soon that how these isolates differ. Okay? So phase 3 trials, that was my job. I monitored them for the World Health Organization for a long time, 20 years. That's me wandering around, going from place to place, looking at what they've done. I'm more or less the coach. I don't like to call it the monitor. I will always remember GCP, GLP, GMP. First, I say, show me your clinical outcomes. Let me see your records, unprepared, randomly. Who did you enter? Who did you exclude? How did you monitor them? Were people vomiting? Did they really absorb the drug? Et cetera, et cetera. Next, I asked them for drug levels, okay? Or if it's a vaccine, I get a sample for antibody markers, yeah? I take that sample and I take it back to the lab to assess what was the drug level in the patient who cured versus the patient who failed. It is possible that people who failed, they had malabsorption, they vomited, they had diarrhea. They just didn't absorb. They didn't make the drug. Who knows? I can do this between different places, a lot of cures, a lot of failures, or within one place, there's cures and failures. That was my job. And when we did this between not just places, but time, I can do it from 1 year and then 10 years later. When we did this stuff, looking at levels in the patients who cured or failed, we published this in Lancet. One of the vaccines makers, their vaccine was no good anymore because it was not making great antibody response. We have published this on drug formulations because they prepared it. It wasn't actually standard formulation, not for horses, but homemade stuff, too much contaminants. No wonder the people got sick. That wasn't the disease, that was your drug, okay? And we can resolve that by pulling drug levels. Also, we pull isolates. You see that traveling? I took back in liquid nitrogen samples for drug and sample for isolates. What's isolate? That's the germ that broke through. You're going to compare in the isolates, this is kind of important, you're going to grow them up in culture or in mice if you can't grow it in culture, and you're going to challenge the mice or the petri dish with the drug to confirm that, oh, no wonder it failed in Brazil. Their isolate is more resistant to your drug, ivermectin. Oh, no wonder it worked in Bangladesh. Their isolate was more sensitive. You can do it place to place, or you can do it at different times. You saw how the variants change in Brazil. Do you think it didn't work in Brazil when it was pink, and maybe now it works? Now that it's purple for whatever, Omicron or something? Of course, the variants change, okay? But you didn't draw the isolates, did you? So it's only speculation to then, okay? Now, this is the rule. In general, when you find a drug that works or doesn't work, and you travel the world, you will see differences. Cures a lot here, fails a lot there. So in malaria, when we found a drug that worked, we found it worked generally in malaria. We look for resistance in the 3 areas. The hottest area is Southeast Asia, then it's Central America, South America, Latin America, then it's Africa, the most sensitive. So when we first tested it that it worked in Southeast Asia, immediately they tested it in Brazil to make sure it works there, and then Africa. And it worked like a charm in Africa until 10 years later when resistance came. It's understood that simultaneously you get different results. That's interesting. And also, if you don't believe that it goes beyond malaria, this is Chagas disease, a parasitic disease at that time we worked South America. Within 3 places, northern Argentina, kind of south central Brazil, and eastern Bolivia, all within a small area, there were claims that an off-label drug, allopurinol for gout, you know the safe dose, right, was curing or not curing Chagas, okay? The Argentinians found that it works, but the Bolivians and Brazilians, I monitored this study, said it doesn't work. You draw the isolates, you give it to the mice, they had trouble culturing it. The mice is an animal culture, and you challenge it to the drug to confirm. And in fact, it was true. The Argentinian variant was very sensitive, and the Brazilian and Bolivian weren't so sensitive. But you do not say that one group of successes disproves the other. It's standard. This is standard. Tuberculosis, drug-resistant tuberculosis. Don't you think some places in the world have it and some don't? Don't you think it changes over time? Yes, it does, et cetera, et cetera. MRSA, everything. So this is my last slide. In summary, when you see failures and successes within the place or between different places in any kind of study for curative, right? Phase 2, 3, 4. These are the ones that infection. Try to think or complete the following. Confirm the source of the drug and the storage, the drug formulation. Make sure they don't use veterinary drug or homemade drug or adulterated drug. Drugs are so expensive overseas, they will cut it to give half the dose. Oh, by the way, that's why we trouble with expensive drugs. They cut it, they do work, okay? But often we were giving the lowest curative dose, so why would you cut it? Because now you're only going to cure half the people. Okay, but you get it. The therapeutic index is important. You will get the drug levels on the subjects as well as the source in your liquid nitrogen container, and you're going to compare the successes and the failures. Okay? You should get the isolates, the germ, for lab testing. Yeah? They're going to grow it up in culture, and you're going to test it against the drug after the fact of failure or success. And if that doesn't work, maybe you should do the metabolite. The drug itself isn't active. It's only when it's taken into the body, the metabolites that your body makes it are more active than the drug. We got burned a little bit on this. The drug doesn't seem to work. Well, not in vitro it doesn't, but when you pass it through a human or a rat, it makes an active metabolite, okay? And you can compare successes and failure. But those of you who are listening say, wait a minute, it cures in a bunch of people. How do you get the isolate? It cured them. You get it before it cured them. You get it on everybody, and when it's cured, then you test the one that it worked against, okay? I mean, it's a subtle point, but some people get upset. You're gonna take all your clinical laboratory and the GMP, GLP, GCP, and combine it all and give me some answer why you think it works and why it doesn't, either within a study or among different studies. And you're going to read what other people have done to address this issue. TB, malaria, Chagas disease, whatever, okay? So, if you think I'm just making this up or I haven't published it, here's two neat publications we produced, okay? This is in Brazil, Journal of Infectious Disease, about 23 years ago. The drug worked fantastically, cured almost everybody in our study. One or two people, I think it was one guy, failed. Okay, so what's his problem? Just dumb luck? Well, a lot of people said, well, it's not going to cure everybody. Why did he fail? I'm curious. So we did that. We drew his serum level and we drew his isolate. His serum level was kind of low. It wasn't zero, it's kind of low. But his isolate was pretty high. It wasn't off the chart like Southeast Asia. This is Brazil, Southeast Asia, but it's kind of high. And so we said, you know, I think there's a resistant strain in Brazil already to the drug, and it's going to pick off the guys with low levels. And lo and behold, just on one failure, we published in GID, a good journal. If you want to see me use dose response, here is our publication in 2001 out of Brazil. Things are failing. We simply took the pill, number of pills divided by the weight, a phase 2 study, trend analysis, the guys with the lower dose, the heavier people, failed. It was repeated for the next 5 years. It went to a phase 3 study and confirmed what we saw. That's the end of the clinical presentation. I have been challenged, or I'm inspired by the Scandinavians, Sweden, Dr. Bath. He always presents his complicated issues with fish. Me too, I'm a fisherman. So I'm going to give you an example of what I just said with fish. Okay. Suppose I sell fishing gear. I sell it, the pole, the line, and the reel. And I tell you it works real well, okay? Some guys go and fish with it and they say, this is junk, I'm not catching anything. Other people say, this is terrific, okay? Maybe Brazil, maybe Bangladesh. Well, why do people catch and don't catch? It's my same tackle, line, reel, and pole. Well, you would expect it, right? If you're catching different fish, like the green fish versus red fish. Oh, Dr. Pang, this is so obvious it's insulting. Really? Tell me about the Brazil study again, where the phase 3 study showed lots of failures, but the Bangladesh and then 6 other studies showed success. Did anyone draw isolates? Isolates are like saying, hey, what kind of fish are you catching? Okay. Is that the end of the analogy? No. It's this study here that takes it a little more. GCP, GMP, GLP. Within one study, check this out. You're only catching one fish. What do we want to focus on? The green fish. You're in an area at a certain time, and you're only catching one fish. And a bunch of people, you give your rod and reel and deer and all to 100 people, and 5% caught nothing. Oh, so it's failing in 5%. Isn't it? Is anyone curious why it failed in 5%? Well, it's just dumb luck. That is a phase 3 trial. Statistics. Oh, it's just dumb luck. You don't expect things to work all the time. That's a simple kind of cop-out. What you should do is interview the 5 people carefully, 5 people who caught nothing, and you should pull maybe, I don't know, 15 people at random who caught something. Maybe who caught the most. I don't care who caught something. Hey, you're all using the same rod, reel, and line. So what's your problem? Well, how much bait did you put on? Oh, by the way, I brought the bait, so everybody's using the same bait, but I didn't tell them how much to put on, okay? So, maybe they put on different amounts of bait. It's pretty obvious. If you put no bait, you caught nothing, okay? Fine. Let me look at your hook. You're supposed to sharpen the hook. Maybe your hook, it varies amongst 100 people. What's dull as dull can be. You supplied the hook. I just gave the pole, the line, and the reel. We all agree that a dull hook catches nothing at all. The extremes. So maybe if you got no bait, you catch nothing, regardless of your hook. Maybe if you got a dull hook, regardless of your bait, you catch nothing. But what if the bait is kind of low and the hook is kind of dull, and you guys caught nothing? You see, it's not all or none. A little bit bad bait, low bait, a little bit dull hook, or the right combination, you 5 caught nothing. Isn't that this study right here? The guy didn't have 0 drug. The guy didn't have the highest resistant. He was number 2 in the resistant pattern, but he was number 3 in the lowest drug level. And it's the combination. It's not all or none. And isn't that how drug resistance comes in first? 2 ways. It's either imported, here it is right there, high resistance, or it slowly evolves in your site, picking off the lowest levels first. That's why we take out antibiotics from the soap, so we don't slowly foster the evolution of drug resistance. So I leave that to you. Nothing is simple. The fishermen kind of know it. We just don't know how to express it. Thank you.